In this session, you are going to listen about the commonly asked RBC practical viva questions with answers. Let us begin. First one, which diluting fluid do you use to calculate the RBC? Answer, Heinz fluid. This is the ideal fluid for diluting the blood because it is isotonic. Second question, tell the composition and functions of the Heinz fluid. Composition of the Heinz fluid. Sodium chloride 0.50 grams. It maintains the tonicity. Sodium sulfate 0.25 grams. It acts as anticoagulant. Mercury chloride 0.25 grams. It acts as antifungal and antimicrobial agent, prevents the contamination and growth of the microorganisms, and distilled water is 100 ml serves as a solvent. Third question, what are other solutions we can use instead of Heinz fluid? Daisy solution. This diluting fluid is an alternative to the Heinz solution. It is simple to prepare and keep for long time. It contains 3.13 grams of trisodium citrate, 1 ml of 37% formalin, and distilled water about 100 ml. Another fluid also we can use for the diluting the RBC, that is the normal saline. 0.9% sodium chloride solution we can use instead of Heinz fluid and Daisy's fluid, but it is not advisable because there is a likely formation of the Rowlex wheels to be observed. Fourth question, how much diluting fluid do you take? 0.5 parts of the blood we used to take, and then we used to take a diluting fluid up to the maximum mark, that is the 101. Fifth question, formula for the diluting factor and how much is the diluting factor in RBC count. Coming to the formula of the diluting factor, the final volume achieved by the original volume of blood has taken. Then the diluting factor in RBC count, that is 1 in 200. What is the next question? What is the final formula in RBC count? The final formula in RBC count, that is N into 10,000. Next question, what are the common sorts of errors we can observe in RBC count? There are three types of errors we used to observe commonly. First one, pipette error. Second one, chamber error. Third one, field error. Next question, what are the functions of the bead in the bulb? The red bead present in the RBC pipette helps in mixing the contents of the bulb thoroughly, and also it helps in identifying the pipette at a distance, and thirdly it tells whether the bulb is dry or not. Next question, how will you identify the RBC pipette? RBC pipette we can identify first color of the mouthpiece, it is red in color. Next, bulb size, it is a bigger and compared to the WBC pipette. Next, it consists of red color bead. Next, coming to the markings on the pipette, the maximum mark that is present in the RBC pipette, that is 101. In case of WBC pipette, that is a 11 mark. Next question, how you can say whether the pipette is dry or wet? The pipette is dry, the bead in the bulb is freely moved. Otherwise, if it is a wet, bead used to be attached to the walls of the pipette. Next question, how will you charge the chamber? The tip of the pipette used to be touched at the junction between the covered slip and the Newbar's chamber. If we press the tube, solution will enter into the gap between the covered slip and surface of the Newbar's chamber. Why you should not use chipped or broken covered slip? A chipped or broken cover slip will not cover the floor piece properly, that is resulting in uneven distribution of the cells. Next question. How will you differentiate red cell from the dust particle? Dust particles are large in size, irregular in shape. If you use the fine adjustment, they will become too dark. If it is a RBC cell, those are refractile and at the center pale we can observe. Next question. Can we see WBC in RBC counting? Yes, an occasional leukocyte may be seen. It seems to be larger than red cells, appears refractile and granular. It also contains a nucleus. Next, its ratio of WBC to RBC is 1:600. Therefore, hardly any WBCs are counted. Next question. Which power of a lens you will use for the RBC count and how you will adjust? 40x objective piece we have to use for the counting of the RBC. Adjustments for 40x. Condenser must be in the middle position and we have to open the iris diaphragm slightly. Next question. What are the rules of the counting? Why we should follow them? We can use either L method or inverted L method to count the RBC to avoid false positive and false negative results. Next question. What are the normal RBC count? In males, 4.5 to 6 million cells per millimeter cube of blood. In females, 4 to 5 millions per millimeter cube of blood. In newborns, 6 to 8 million cells per millimeter cube of blood. What is the lifespan of RBC? It is about 120 days. What is the fate of RBC? The worn out RBC's fragments in the circulating blood. These are old cells removed and destroyed by the phagocytes in the spleen and the liver. Next question. What are the physiological variations in RBC count? The decreased RBC count, physiological conditions, pregnancy, in children, females have lower RBC count than the males. Pathological conditions, anemia, hemodilution conditions, in excess ADH secretion as seen in posterior pituitary tumors. Increase in RBC count, physiological conditions, high altitudes, newborns, hemoconcentration during excessive sweating. Pathological conditions, heme concentration in diarrhea and vomiting, chronic hypoxia in congenital heart diseases, chronic obstructive pulmonary diseases, and polycythemia vera. Next question. What is erythropoiesis? The formation of RBC from the bone marrow stem cells. What are the sites of the erythropoiesis? In fetal life, spleen, liver, thymus, and bone marrow. In infancy, red bone marrow. In adult life, long bones. Next question. Do you know any other accurate method for determination of RBC count? An accurate and more reliable method for counting RBC is by way of electronic cell counter, in which error is only about 2%. Next question. What is the difference between polycythemia and polycythemia vera? Polycythemia is a condition in which RBC count increases above 6 millions per cubic millimeter of blood, whereas polycythemia vera is cancerous condition of bone marrow which causes marked increase in the RBC count. Next question. What are the other functions of RBC pipette? Answer. Other than RBC counting, RBC pipette we can use for sperm count, platelet count, and WBC count during leukemic conditions. Next question. Why in females RBC values less than the males? Answer. It is due to the effect of estrogen and androgens on the erythropoiesis. Thank you for listening.